Mentoring is described in the literature as an essential component to career success, productivity, and satisfaction. Hello and welcome everyone to today's workshop on project-based bioinformatics and data science training for faculty and mentors. This workshop is for faculty seeking to examine, share, and strengthen their own mentoring practices. Faculty will learn evidence-based approaches to research and conceptual and practical knowledge about various specialization tracks, which include precision medicine, infectious diseases, neurosciences, oncology, space omics, agriculture, and AI-based drug discovery. So before we begin with today's workshop, let me introduce us. We are a US-based bioinformatics company who is working with multiple academic and commercial collaborators to develop easy-to-use analytical tools, and our mission is to make bioinformatics more accessible. The training has been completed by over 15,000 participants from 162 countries in over 300 workshops. And due to this fast growth, our team is working with local and regional coordinators that are helping refine the local program logistics and leverage our online training resources, adapting them to the needs of students and researchers around the world. I'm honored to introduce the founders and team members of the company. Dr. Alfred Tauber comes from a medical background with an expertise in oncology, immunology, and biochemistry of inflammation. And one of his major projects is the establishment of Tauber Bioinformatics Research Center, which has been at the core of what we do. Dr. Leonid Brodsky is the director of Tauber Bioinformatics Research Center and is an expert in bioinformatics and biostatistics. Our platform is a result of hard work by some of the people mentioned here on the right. Julia Panov is an expert bioinformatician. Avi Titovsky leads the development of T-Bioinfo platform. Dr. Mohit Mazumdar is an experienced bioinformatician with multiple publications in peer-reviewed journals. He has been developing collaborations with universities as a mentor and project lead for OmicsLogic training and research fellowship. Dr. Raghavendran Lakshminarayanan is an accomplished computational biologist with an expertise in large-scale analysis of multi-omics data and data science methods in precision oncology and protein DNA structural bioinformatics. Ms. Laura Murray, Educational Program Coordinator, has a background in microbiology and bioinformatics, and her focus and areas of expertise are in metagenomics, transcriptomics, and genomics. And my name is Sonalika Ray, and I'm the Community Manager responsible for daily interaction with thousands of users that are a part of our bioinformatics community, which is managed by Biotech. So let's discuss about bioinformatics today. What is bioinformatics? It is the development, implementation, and usage of methods and software tools for understanding biological data. Bioinformatics has practical applications in healthcare, agriculture, and mitigating climate change. As this field progresses, bioinformatics poses several challenges for both students trying to learn it and faculty trying to teach it. In this presentation, I will explain the current challenges and limitations in bioinformatics, describing how we aim to address these challenges with the Omixlogic platform, describe Bloom's taxonomy and how it can be used in bioinformatics coursework, and explore the OmicsLogic program and how it utilizes Bloom's taxonomy in structuring its course, pipelines, and projects, and finally describe several outcomes of the OmicsLogic program. So what are some of the challenges that the field of bioinformatics is currently facing? To begin, this field is constantly growing and developing new tech and software to improve processing time and power. The data sets that are analyzed are usually very large and take immense amounts of computing power and memory that many personal computers do not have. Also, despite being publicly available, data are also usually complex to access, and coding knowledge is usually required to do so. Similarly, analysis pipelines and programs require coding knowledge that many biologists do not have. In a university setting, students have the opportunity to learn the background information of designing and implementing a bioinformatics project, but do not receive practical hands-on experience with the full workflow due to the limitations mentioned previously. Similarly, faculty faces challenges with being able to cover a diverse range of topics if they are specialized in a certain area of bioinformatics or if they are unfamiliar with bioinformatics in general. For example, a professor whose research focuses on oncogene expression might not know much about metagenomics of seawater. To overcome these challenges, the Omixlogic platform was developed. This program aims to standardize, organize, and systemize bioinformatics training in order to make these analyses usable for any biologist or clinician. Some problems for standardization. Materials for these courses are usually developed in-house, taught sporadically, and are rarely updated. Organization. Because the challenge is to combine several different topics together, the flow and transitions are needed to be developed. Systemization. How can you know that someone is stuck and needs help or allow someone to run forward? OmicsLogic uses standardized modules and interactive sessions beginning with mastering terminology and concepts to implementing these concepts with real data sets in a project-based approach. Learning process. In order to create a clear path of skill development, OmicsLogic employs the Bloom's taxonomy levels of learning. This approach starts with the basics. Students recall facts and terminology and are able to repeat and memorize the material. With this base, the learning builds to conceptual knowledge, being able to understand concepts and apply information to new situations. The student will then be able to analyze data by drawing connections and evaluate when to use certain methods. This leads them to the highest level of learning, the ability to design and create original work. So how do OmicsLogic resources help in advancing bioinformatics teaching? OmicsLogic is a portal for practical and theoretical learning of bioinformatics. Composed of courses designed to take you through the theoretical knowledge, OmicsLogic takes a combination of training modules, data analysis tools, curated project datasets, and interactive sessions with mentors to give a student a clear path to the Bloom's taxonomy pyramid. No matter what the user's initial experience with bioinformatics is, OmicsLogic is a useful tool for gaining a baseline knowledge of the theory behind bioinformatics, analysis of large data sets, and introduction to basic coding languages like R and Python, or beginning a data analysis project. These 6 courses were taken from the core OmicsLogic curriculum, which is available online, beginning with an introduction to the application of bioinformatics and an overview of data types. The student is able to learn terminology and factual knowledge to build upon. From there, genomics, transcriptomics, metagenomics, epigenomics, and data science are introduced. Each course starts with important terms and concepts and then introduces case studies and pipelines related to each one. This allows the students to gain the conceptual knowledge in order to begin their own projects. Expanding basic curriculum with projects. Bioinformatics methods are best learned in the context of real case studies which are prepared by our network of mentors and collaborators that illustrate how various bioinformatics methods and omics data types are used in current research in medicine, biotechnology, agriculture, and even astrobiology. The projects are prepared by taking publicly available datasets and using them to explore and understand both the research challenge and the analysis path to extract meaningful insights from complex omics data. The OmicsLogic platform gives students options for analyzing a given dataset. These datasets and associated publications can be used in several ways. You can learn bioinformatics without any coding at all. For that, in each project, we explain the analysis pipelines on the user-friendly tBioinfo platform for big data analysis. You can also learn the scripts behind many of the steps, learning to wrangle, clean, visualize, and analyze data in popular languages like R and Python in order to create your own analysis. Finally, you can hear about the projects and their research applications in our mentor-guided programs where experts share their experience and support research projects developed by you. Here is an example of the code-independent pipeline. The pipeline builder interface on the T-Bioinfo platform is a color-coded pipeline that helps establish a logical connection between the technical names of data analysis methods and the logical steps needed to build the full pipeline. Data is put in an input file, and then the analysis can be chosen based off of the type of research. Each analysis is separated into three colors: blue for pre-processing, red for mapping, and green for quantification. Each time you click a button, a pop-up will contain additional information useful for building the pipeline. The ease of the color-coded buttons allows programs to be run without any coding knowledge. After using the T-Bioinfo platform to visualize the pipelines and practice analyzing, students are then able to take their theoretical knowledge, remember and understand levels of blooms, and apply that in a project. Apply, analyze, and evaluate levels of blooms. Similarly, faculty should be able to assign the OmicsLogic program to their students with the understanding that it will give them holistic and comprehensive knowledge of bioinformatics, beginning with bytes and molecules and on to designing their own question-based project. Another goal of the project is for students to be able to read primary literature and understand and recreate the methodology. This leads to the ability to encounter errors, apply theoretical knowledge of why those errors are occurring, and find a solution. OmicsLogic employs a hybrid approach, combining learning and research. To ensure that students are building off of the first level of Bloom's taxonomy, OmicsLogic tracks progress with activities like pipelines, coding, and literature reviews, quizzes on topics, key terms, and important questions, and covers a broad range of topics like biomedical research, biotechnology, and agriculture. Then students are able to continue their knowledge base with a research assessment, making sure they are able to understand methods, challenges, and results, as well as being able to comprehend primary research literature. They should also be able to ask questions based on data exploration. Bioinformatics combines life science terms with data science skills, which allows researchers to answer life science questions with reproducible data analysis methods. OmicsLogic research fellows are able to ask research questions about any bioinformatics topic, leading to a diverse range of projects, from analyzing affected biological pathways in multiple sclerosis to brain metastasis due to small cell lung cancer. These students are able to apply what they have learned in the coursework, work with a group of professional mentors, and submit their projects on the portal for review. Students should then be able to reach the highest level of Bloom's taxonomy, create. This is achieved through the creation of their own analysis project. For example, Sime Dolaner, an undergraduate researcher at Bahçesaray University, Istanbul, Turkey, who finished her OmicsLogic research training, was able to work on the project involving chronic lymphocytic leukemia, CLL, a type of blood cancer that has a very heterogeneous biological background and diverse treatment strategies. Here at first, she elucidated 870 significantly differentially expressed protein coding genes that were involved in the biogenesis and processing of RNA. Consequently, these findings led our study to investigate non-coding RNA. 33 long non-coding RNAs were found to be significantly differentially expressed among two conditions based on differential gene expression analysis. One final example of a learning outcome of the OmicsLogic program is this project presentation for a project done in collaboration between Noguchi Memorial Institute for Medical Research and Pine Biotech. Here is our team at Pine Biotech who were involved with the project: Dr. Mohit Mazumdar, Dr. Harpreet Kaur, as well as our collaborators at University of Ghana, Dr. Anita Gansha and Dr. Benedicta Mensa. This poster was on Plasmodium falciparum apical membrane antigen, rupture neck protein 2 binding, which is a critical interaction for Plasmodium falciparum merozoite invasion of human host red blood cells. Several proposed malaria vaccine candidates designed to prevent such binding and limit merozoite entry have been studied with variable success at in vitro and early clinical trial stages. By performing haplotype network analysis of genomic sequencing data from sites with variable infection rates, comparing our data with malaria gen repository from another region in Ghana, and mapping of genomic variants onto the protein structures, we arrive at preliminary conclusions about local variation and immunogenic epitope affecting variants for consideration. In this poster, we share preliminary findings that suggest that such genomic data can be used to develop a local strategy for efficacious vaccine design against Plasmodium falciparum blood stage infection in Ghana and surrounding regions. Omixlogic is proud to collaborate with diverse programs, for example, NEPI, the National Association of Idea Principal Investigators. We also work with over 15,000 students at community colleges like Glendale Community College, top-tier universities like Georgetown University, as well as 20 universities around the world. Now to talk about the programs in